Hello, I'm Steve Goodman. I'm the Vice Chancellor for Research at the University of Tennessee Health Science Center. And today we're going to present the exciting things that are going on in the state of Tennessee concerning stem cell therapeutics and regenerative medicine, from basic research to clinical trials. You'll hear about the Tennessee Institute of Regenerative Medicine and the Clinical Trials Network of Tennessee, which is also referred to as CTN2. And you'll hear how they are related. So our first speaker is going to be Dr. Gabor Tigyi, and Dr. Tigyi will be talking about the Tennessee Institute of Regenerative Medicine.
Thank you, Steve. Good morning, everybody. It is my pleasure to introduce you to TENERM, the Tennessee Institute of Regenerative Medicine. You will hear from me and also our Chief Scientist, Dr. James Kang, following my presentation. We'll be focusing on the scientific advances that we are pursuing at TENERM. How was TENERM born? We got formed in 2017 by the Memphis Research Consortium, which is an alliance of the University of Tennessee, St. Jude Research Hospital, the University of Memphis, and also representatives from major local businesses and city leaders. Memphis has strong medical industry base, including Medtronic, Smith & Nephew, Stryker Medical, and the international shipping giant FedEx, interested in global biologistics. A partnership was formed to develop regenerative medicine in coordination with the three major research institutions. With an initial investment by the state of Tennessee, the Memphis Institute of Regenerative Medicine was launched, that is now rebranding as the Tennessee Institute of Regenerative Medicine, TENERM, in recognition of several other research groups joining from across the state of Tennessee. There are four research groups in our center, institute, and these are the following four. Number one is 3D biofabrication methodologies and manufacturing that is largely centered at the University of Memphis. Number two is cell interactions with 3D bioprinted blood vessels, basic and translational approaches, that is led by Dr. Kang and primarily run out of the University of Tennessee Health Science Center. Number three is focused on stem cell enhanced tissue regeneration, engineering of vascularized bone cartilage grafts from adipose derived stem cells, and dental stem cell based technologies led by Dr. Kang and George Huang. The fourth direction of research is aimed at harnessing hematopoietic and cancer stem-like cells for regenerative medicine, and it is a group that is composed of investigators at St. Jude Children's Research Hospital, led by Dr. Mitch Weiss, and two of us at the University of Tennessee Health Science Center. I'd like to take you through these four research directions that are being pursued. Biofabrication of implantable, injectable, and printable hydrogels for tissue scaffolds and delivery devices is the first direction under the first project. The next direction is basically focused on nanofiber additive manufacturing for tissue regeneration and for other medical devices. The third direction of research is using magnesium-based alloys that are biodegradable and can be shaped to mimic orthopedic devices and also guide bone regeneration when functionalized with stem cells. As I mentioned, these are primarily pursued by the University of Memphis in our city. Number two is the cell-cell interactions with the bioprinted vessels, basic and translational approaches direction. And this group aims to understand the regulation and underlying mechanisms of blood vessel regeneration by using Revotech company's REVOVAS 3D bioprinted vessel technology. Our overall goals are to improve clinical applications of the novel 3D bioprinted stem cell vascular grafts and to open up additional opportunities for regenerative medicine research. Three areas are being pursued. The first one is to develop a mouse model of vascular implantation using stem cell derived 3D bioprinted blood vessels. This opens up our investigation to a lot of genetically engineered mice and other benefits of the mouse models. Number two is to identify critical gene variants that cause differences in blood vessel regeneration. And the third one is the definition and validation of gene variants that control regeneration under normal conditions. Let me show you a couple of examples of this technology. First is the left side shows this machine that we call the biosynspherer. This instrument is a microfluidic bioink generator, which has been now scaled for small animal applications. And it generates these biosynspheres that you see here in the middle, pointed by the cursor. And these stem cells are these little white dots, but we have labeled them with red fluorescence, and you can see them that they are sitting here in this artificially generated stem cell niche, which is this magic ball, the biosynsphere. There is a cortex, which is labeled with green fluorescence. This is functionalization, and it allows the targeting of these biosynspheres to various places in the body. The lower right panel shows the prototype of this 3D bioprinter that was developed for vessels that we find in mice. And here's an example actually of the surgery of implantation of a bioprinted mouse aorta. Here's the distal portion of the mouse aorta, and it is now cut out and replaced with this bioprinted vessel graft. And here is the end of the surgery. The clips are removed and the blood flow is restored. I'd like to note that the outer diameter of this vessel is 1.2 millimeters, and the inner diameter is 0.8 millimeters, which is smaller than a human coronary artery. The third research interest of TENERM is engineering of vascularized bone cartilage graft from adipose tissue-derived stem cells, and also dental stem cell-based technologies. First, let's look at the osteoarthritis problem. It is a fairly a large problem affecting 32.5 million US adults annually. Restoration of normal bone and bone cartilage integration is an opportunity to apply stem cell regeneration technologies. Now, cartilage breakdown in osteoarthritis increases synovial inflammation, which in turn increases cartilage breakdown. Stem cell therapy, through release of paracrine factors, blocks this vicious circle and can inhibit joint destruction and ultimately repair and regenerate damaged cartilage. The problem, however, is that the injected stem cells do not last long, but a day or two, and do not regrow cartilage. So how can this problem be solved? Here's an example of using the biosynsphere technology to actually overcome this limitation. What you see on the left side is a biosynsphere that contains these adipocyte-derived stem cell. And here on the right side of the slide, kind of in the middle, what we demonstrate is a joint of a rat that was injected with a suspension of adipose-derived stem cells. And what you see here is that these cells were fluorescently labeled, and there is very little fluorescence is detectable 4 days after injection. And if you look at this Eppendorf tube, again in the middle, the fluorescence coming from the isolated and scraped cells spun down to the bottom is very little, and there is a lot of cell debris that floats to the top of the tube. Compare this with the right-hand side, which is the other joint, other knee of the rat, which was injected with stem cells in biosyn spheres. And I think it speaks for itself because you see a lot of red and yellow colors. And also take a look at the Eppendorf tube, and you see that there is a sizable pellet representing a lot of surviving cells 4 days after injection, and there's no debris, so there's very little cell loss. So that certainly is a very promising approach to overcome the limited duration of survival of these injected stem cells. Another application is focused on dental pulp stem cells. The tooth, the top layer depicted in white is the enamel that we always try to keep nice and white and bright. Underneath is the cellular layer, bone-like layer, which is the dentin. This is where we find these odontoblasts that generate enamel and also the dentin itself. And in the middle, like the bone marrow of the tooth, is the dental pulp, which is where the dental pulp stem cells reside. The problem fundamentally is that these odontoblasts are terminally differentiated cells like neurons and do not regrow once the tooth has been formed. So if you have a cavity and you lose your enamel and the cavity goes down to the dentin will not regenerate because these cells are lost and not replaced. So what can we do about this methodology? We have discovered that a lipid mediator called lysophosphatidic acid is critical for maintaining stemness of dental pulp stem cells. This mediator, LPA, is generated by the enzyme autotaxin, labeled as ATX. And this LPA growth factor-like lipid mediator works through G protein coupled receptors, and the LPR2 receptor subtype is the one that is critical for maintaining stemness. We have developed an inhibitor of this receptor subtype called PNUT 001, and we tested it in the following experiment. This is an in vitro experiment. These are dental pulp stem cells in culture, and then they are transferred into an osteogenic differentiation medium, and after 21 days, they accumulate calcium depicted by this red staining. Now, here on the bottom, what you see is the same setting, except that we are adding increasing concentrations of PNUT to the cultures, and you see that within 7 days, we actually reach the same type of calcification that we have seen in the other osteogenic medium at 21 days. So we have sped up the process at least threefold. Now, does it work in vivo? Well, here's a little bit of rat dentistry. We drilled a hole here in this molar tooth of the rat and plugged it either with a vehicle soaked collagen plug or with this PNUT also delivered in the solvent with a collagen carrier. The important thing is shown here on the right on this micro CT images taken from control teeth and PNUT plugged teeth of the rat. And after 6 weeks of leaving these collagen plugs in the cavity, what you see is that the cavity is still open. This white debris shows that the gray area, which is the dentin area, has not healed. It hasn't filled in the gap in these two views. But if you compare that with the bottom one, which is the PNUT treated tooth, you'll see that there's a complete dentin bridge that has been formed. And the main message that I wanted to convey to you with the demonstration of this experiment is that this procedure can obviate the need for root canals and turning the tooth into basically a lifeless piece of bone in our jaw, which will ultimately fall out. So I think this is completely a complete breakthrough in this therapeutic intervention. The fourth group has two projects that we pursue. Project number one is a genome editing of sickle cell disease, and it utilizes a principle that is naturally occurring, that is the fetal hemoglobin that consists of alpha and gamma subunits is switched off at birth, and then the adult type, which contains the mutation that leads to the sickling of the red blood cells, is turned on. And basically, the holy grail of this technology is that we have to block the expression of the adult hemoglobin and maintain the expression of the hemoglobin fetal type, because that can actually increase the probability of survival, as it has been shown for in clinical trials. And the idea is that if it can be maintained at 20%, then it will provide a 90% probability of survival in the patients. So as a result of this hypothesis, our colleagues at St. Jude, with the leadership of Dr. Mitch Weiss, have silenced the transcription factor that drives the expression of the adult hemoglobin, and then took these cells, put it into a mouse, that is a humanized mouse, and 16 weeks later, looked at the bone marrow to see whether these engineered cells took hold. And as you can see, for 3 mice, that is demonstrated here, the blue columns represent the fetal hemoglobin expressing cell population, which is at about 20% or higher for these 2 mice, and it's considerably less for the third one. But certainly this is encouraging, and our colleagues believe that as early as 2022, they will be able to file an IND and start clinical trials using these genetically engineered red blood cell progenitors. The last project in group 4 is focused on cancer stem cells. And it comes back to the same principle that I already introduced to you, that the autotaxin LPA axis regulates stemness. And what we have done is engaged in a drug discovery project for many years now to develop autotaxin and LPA receptor inhibitors. And this is very important because these stem cells are responsible for the growth and survival of these tumors, the metastatic potential of the tumors, and the therapy resistance and the immunomodulation resistance to immunotherapy is all encoded in the cancer stem cell. So just to demonstrate the proof of principle of our studies, paclitaxel is a major chemotherapeutic that we have used to grow stem cells that become gradually resistant to higher and higher concentrations of this chemotherapeutic. And we looked at the expression of the LPA2 receptor, which is proportionally increased. So the higher the resistance, the more LPR2 there is. And then we used these tumor sphere cultures, which is basically every tumor sphere represents or grows out of a single cancer stem cell. And we treated them with 2 drugs. Here's the PNUT1, the LPR2 receptor inhibitor, and the BSA3, which is the autotaxin inhibitor. And what you can see is that after treatment, we have drastically and significantly reduced the number of surviving tumor spheres, that is the number of stem cells, cancer stem-like cells that were maintained in this culture situation. Let me turn it over to Dr. Kang, our chief scientist, who will tell you more about our translational research efforts to take the 3D bioprinted vessels into human subjects. Thank you.
Okay, thank you, Dr. T. I'm James Kang, the Chief Scientist of Tennessee Institute of Regenerative Medicine. Besides my academic service, I'm also the founder and past president and the CEO of REVOVAS company. What I'm gonna do in next 10 minutes also, I will share a major breakthrough on vascular regeneration from 3D bioprinted stem cell grafts and the clinical trial. Next slide, please. Atherosclerosis is a major problem worldwide. Go back. This particular affect the aging population, but our aging population rapidly grow and making this problem even much worse. And one of the problems related to atherosclerosis is coronary artery disease. Once you have this blockage of the coronary artery, leading to heart failure eventually. Cardiovascular disease, as we all know, is the leading cause of morbidity and mobility of world in the whole in the world. And to solve the problem, atherosclerosis injured blood vessels. In the 50s of last century, scientists developed some synthetic vascular graft based on the polytetrafluoroethylene, PTFE material. And this graft can use as a bypass or replace the injured vascular and let the blood reflow. But the problem is after several months or several years implant, the thrombosis will develop and leading to the reblock the vascular system again. To solve this problem, endothelialization of the inner surface of the vascular graft has been the major undertaking of vascular graft application. Next slide, please. Now, tissue engineering technology has been applied to solve the problem endothelialization. The principle is integrate of the cultured cells and scaffold material and make them incorporated together and culture them, eventually hope to generate endothelial cell attached vascular graft. But the problem is this technique never get these endothelial cells evenly or regularly attached to the inner surface of the graft. And also the attached cells never have a good resistance to shear force. About 10 years ago, my group started thinking about using different technology, 3D printing technology, to develop a 3D bioprinting cell graft to solve this endothelialization problem. Next slide, please. Now, at the beginning, we made a big mistake. We used the same principle for the tissue engineering. We think we can just print the cells layers, so use the endothelial cell layer and then a smooth muscle layer and put them together, and then to nurture them and incubate them. Eventually, they become evenly attached, endothelialized vascular graft. This driven design, but it doesn't work. Next slide, back please. So why it doesn't work? Well, we now realize our naturally developed the vascular system is derived from proliferation and differentiation in the embryonic. So if the blood vessel get damaged and our tissue or organ system will recognize the problem, will start its own activation of the repair mechanism and using stem cells to make the repair. But if you don't have a stem cell in place, even you have this injury take place and you have the regeneration, you cannot make this repair fully developed. So if we can supply enough stem cells of the repair material at the beginning of the injury or at the system will start to regenerate or repair its own injury. I call this destination engaged vascular evolving lineage organ regeneration or develop. Now, with this new concept, we develop a 3D bioprinting stem cell graft vascular system. Next slide, please. There are two major components. Would you stop there? There are two major components to make this. First, to make the bio ink of a stem. Secondly, is the printer. Now, you cannot use naked bio ink because in that case, it cannot protect the stemness of stem cells. I cannot ensure the viability of the stem cell in the liquid system. So we need to pack the stem cell in the protective envelope. We call the biosphere. Dr. Tigyi already talked about the biosphere. With this biosphere produced, we collect the biosphere. I go ahead to move. Go ahead. So with this biosphere, we collect it and put it into the ink jar, and then bring to the computer, bring to the printer. This printer is invented by REVOVAS, and on this, you can see with this printer you can print a vascular like a graft using stem cells. And on the surface, we use a synthetic vascular graft to cover the surface of this stem cell graft to make the hybrid graft. And this graft, after certain solidation treatment, and is ready for transplantation. And make this graft only need 2 hours. And this is we implanted the graft into pigs, and monkey use this use their abdominal aorta. So basically, simply cut a piece of aorta and replace it as injured with this graft. And here is you see the blood flow through this implanted graft. After certain days, we experimentally remove this graft and to look at what happened and the graft in the body. Now, this is the blood vessel fully developed from the graft. In the body is pretty stable and relaxed, and so it's have very good stretchability. And next, please. If you look at historically, after you remove the vascular, this is a vascular structure by the histological examination. What you see on the top, this is a vascular system or the structure developed from a 3D bioprinted graft. And basically you can see they are identical to the normal vascular structure, including endothelial layers and smooth muscle composition and collagen deposition. Next slide, please. How long this graft can develop into fully functional vascular system. This only need to take 5 days. Basically, as I mentioned, this just reactivate our embryonic stage tissue developer program. So at the 5 day, you can see all of these endothelial layers and also this smooth muscle composition and collagen all formed regularly and biologically. And at 70 days, we look at basically this fully developed vascular system is the same as the normal vascular system. Now we have been using mouse model and I'm sorry, using a monkey model and also pig model for doing this for 7 years, and some monkey has been lived there for more than 5 years, and we do not see allergenic reactions. The only thing we need is anticoagulant that use heparin after the implant within the first days of development, because there's some blood plaque there. After 5 days, and you don't need to do anything, you don't need to use anticoagulant at all. And basically, that's a normal development. We do not see immune response. And as I said, we have been leaving this animal for more than 5 years, and they never develop tumors, so there are no tumorigenesis. So we already use this for human clinical trial. Next, please. So we're so excited about this development and this breakthrough. In the early 2017, so we are contacted with the FDA, discuss with them the possibility of using this newly developed 3D bioprinter stem cell vascular graft we call the REVOVAS for clinical trial. And with the several dialogue with FDA and with their guidance and their help, we finally submit our application, the full and formal application in November 2018, and further they help us to develop a clinical proposal, and we developed the clinical site. And so during that time, in the 2017, this Revotech under my direction and interact with University of Texas Health Science Center with Dr. Steve Goodman, and we discussed, this is the time we established Memphis Institute of Regenerative Medicine. And through this institute, we further developed this blood vessel for the clinical trial, and we have produced this 3D bioprinter vascular system in the Memphis campus and in UTHSC Medical School Center. And also, we are already fully prepared for the clinical trial. We all know, because of the coronavirus pandemic, and this has been delayed. But I expect, and also our team expect, after July 4th, we will reopen this discussion with the FDA, and we will submit our final approval proposal for FDA, and I expect we should take this clinical trial soon. So I would expect the world first 3D bioprint stem cell vascular graft clinical trial would take place at a CTN2 size sometime in October. Why CTN2? I think we should hear from Dr. Steve Goodman talk about how CTN2 can help us or help the world to do clinical trial for biological products.
Well, hello again. I'm now going to talk to you as the president and CEO of the Clinical Trials Network of Tennessee, and I'm going to be sharing this talk with Phil Sestero, who is the executive director and treasurer of CTN2. So CTN2 was created about 3.5 years ago as a 501c3 subsidiary of the University of Tennessee Research Foundation. It's governed by a board that represents CTN2's growing network of hospital systems and large practice plans, as well as representatives from UTHSC and UTRF leadership. It's proven in its first three years to be extremely attractive to pharma and device manufacturers. We are capable of contracting for studies as a 501c3 at the rate that industry requires. We are also capturing research credit for UTHSC that has been missed in the past. Based on the success of CTN2 that you'll learn about, our chancellor at UTHSC has extended our funding into fiscal year 2022 with another $1 million of additional funding beyond that for completing some stated objectives. CTN2 is a statewide clinical trials network, as you will learn, but it's a part of an integrated set of clinical trial entities that are statewide. One is CTN2, another is the Clinical Trials Governance Board, which has leaders from clinical trial offices around the state. Another is our Clinical and Translational Science Institute, which is statewide. And as you will learn, we have developed a statewide enterprise data warehouse behind the UTHSC firewall. We started with only 5 partners that were all located in Memphis. As you see in this slide, over 3 years, we've grown from 5 to 13 hospital and practice plan partners that cover the entire state of Tennessee. In addition, we are currently talking to 5 additional sites that will be shortly joining our entity. This shows you the rapid growth from the beginning, where we started at 5, and then in fiscal year 2019 grew to 8, then 11, 14, and we plan by next year to have at least 16 sites around the state of Tennessee. I'm now going to turn this over to our executive director, Phil Sestero.
Thank you, Dr. Goodman. And in addition to providing credit to the university, research credit to the university, CTN2 provides a number of benefits to our site. First of all, the UTHSC's EDW, which is made up of approximately 2.1 million unique patients and represents approximately 35% of the population in Tennessee and continues to grow. The EDW and the expertise and relationships within the CTN2 leadership team has brought in over 200 new study opportunities to UTHSC and our site's faculty and investigators. Additionally, CTN2 provides extremely flexible study models. We can work with our investigators on studies that they have initiated and found, or studies that we found that could eventually become network-based studies across CTN2. Next slide, please. Another benefit is CTN2 sits with each of our sites upon bringing them on and generates standard operating procedures. These are jointly developed. We're really documenting the processes of our sites while providing some recommendations or ideas on some process improvement. Our sites have really appreciated this. Many of them did not have all of their operating procedures documented, so get them documented and then to start utilizing the procedures and seeing how they were doing towards them. Another benefit that we provide is what we call expert boards. These are small groups of physicians or investigators that are working and researching in a specific area. It's not to be more specific than that. These boards get to meet about potential study sharing or opportunities. They also are a vehicle to provide mentorship to new or junior investigators who are interested in building their CV to be on or become principal investigators on a study. And we have found these to be extremely valuable to our network. And for example, in our first board meeting ever, expert board meeting, excuse me, one of the investigators shared a study that was then opened at another site. And it just shows, as if you create the vehicle for them to talk, they're going to share ideas. Investigators want to collaborate, and we're very proud of that. Lastly, we have a fiduciary responsibility. We handle the flow of money at CTN2 for our sites. So all of the money paid by sponsors comes through CTN2 and then flows to our sites. So setting up, very, very strong financial and accounting processes was very important to us. We've done that. We are audited annually by an independent accounting firm. And to date, we have had no findings, and we're extremely proud of that. Next slide, please. Our contract growth, you can see in the green bars that each year since our inception, we have doubled in our contract value. We expect to at least double next year, and then beyond that, we'll increase by about 4 million each year. This is just through a combination of providing more study opportunities, enhancing services that are available to our sites, and adding additional sites. And so we're really, really proud of the curve that you're seeing there in the upward trend for contract value. Next slide, please. And then number of contracts. And so obviously when you set up a network, it starts out slowly as you're putting a lot of agreements in place and bringing on the sites. But you can see the growth over the last 2 fiscal years for us has been significant. That number continues to grow, and we expect it to grow exponentially going forward. Next slide. So some highlights in our first few years of existence, as Dr. Goodman pointed out, we have 13 sites, 5 of which was the initial plan. So we far exceeded that objective. The expert boards, we brag about these a lot because we're extremely proud of them and the opportunity they provide for network-wide collaboration at CTN2. And many of you understand this, but put together a network is a lot of legal agreements. There's a lot of negotiation and understanding to put a lot of hospitals and physician practices within the network. And so this was a metric that we were kind of surprised when we saw it, that we had put together 33 legal agreements with our sites over the course of our first 3 years of existence. As I've mentioned, 200 plus study opportunities. I think we're going to have to change this slide eventually because I think it is getting close to 300 now. So we're very proud of the relationships we have with our pharma and medical device sponsors and study opportunities that they provide to our investigators. $7.3 million in cumulative research contract value. That's the increase that CTN2 has provided since its inception. And as we showed in previous slides, we only expect this to grow and continue to grow. And no audit findings in our financial audits. Again, extremely proud of that. And then as part of the Chancellor at the University of Tennessee Health Science Center engaged WCG to do an analysis of industry-sponsored clinical trials at the university. And the feedback from that report was extremely positive to the leadership and to the business model. If we can go to the next slide, please. Here are some of the quotes that came directly out of that report that from the WCG, and they saw CTN2 as a beacon for industry trials at University of Tennessee. They mentioned again the leadership and the seasoned research experience and professionals that we had. And they talked about it, and I love these words because it said that had the potential to transform clinical trials administration for the university. Those are great words to have when you're putting something together. And then lastly, they recommended investing more money in CTN2, growing CTN2 and growing the services that it can provide to its investigators across the network. Next slide, please. And so what's coming? We've been very successful in our first 3 years, but what else is coming with this? We've created a regional site development model. And so Tennessee is a very long state, and we're expanding beyond Tennessee additionally. And so how do we do that? And so we have now on staff regional medical directors who are cheerleaders in each of the main cities that we have sites in. They're there talking about CTN2, answering questions, and also helping direct us to new practices where investigators are interested in growing their research. We're going to continue to expand our CTN2 expert boards, and with both additional boards and with the existing boards adding capabilities for them. We're in the process of selecting a clinical trials management system that will be happening any day now. And we're really excited to have that deployed over fiscal year 2022. And this will allow us to aggregate the various clinical trial systems that are out there and provide a centralized reporting and metric capability across our network to make it much less manual than it is now. Lastly, we are opening an office in East Tennessee. Our current office is in West Tennessee. And so opening an office over there just to expand our capabilities and our reach throughout the state and beyond. And so we're very much looking forward to that and hope to have that open by the end of this summer. And so all of this is to position UTHSC as the go-to organization for industry-sponsored trials and improving healthcare, because at the end of the day, it's about improving the healthcare and the capabilities to treat the patients of Tennessee. Next slide, please. And I'll hand it back to Dr. Goodman.
So in summary, CTN2 is an existing organization with a proven success in contracting for clinical trials efficiently. We contract in days compared to universities that usually take months to do clinical trial contracts. And because of that, we've been accepted by institutions and sponsors who appreciate what we bring to the table. We're backed by the power of a UTHSC enterprise data warehouse with 2.1 million potential subjects to attract studies. And even though we started in Memphis and grew throughout the state of Tennessee, our board has given us permission to do clinical trial contracts outside of Tennessee. So we're open and interested in receiving study proposals and inquiries from sponsors and investigators alike for stem cell and regenerative medicine trials, whether they are conducted in Tennessee or within the US or around the world. Any proposals received by us will be promptly vetted. So for all of you, investigators, institutions, industry sponsors, we are open to you. If you want to receive more info related to CTN2 and how we can assist you, please contact myself or Phil at the email addresses that are given. So I hope that you have enjoyed our explanation of these two exciting entities that we have built over the last 3 to 4 years in the state of Tennessee. Thank you very much.